Let's discuss the amination in detail now, means the urea cycle. So as we have discussed that there is carbon dioxide and ammonia. So they collectively make, so let's write ammonium ion. They actually make carbamyl phosphate with the help of same enzyme. Sorry, it's phosphate. Like the enzyme would be carbamyl phosphate synthetase. So it's very easy to learn the name of the enzymes because now the enzymes are almost similar to these one, whatever the product is. Okay, this carbamyl phosphate now join with ornithine. That's why this cycle is named as ornithine cycle as well. So now it is converted into arginosuccinate. Now here, if arginosuccinate is forming, then the enzyme would be like the same one, arginosuccinate synthetase. I'm writing the short form. You don't need to write the short form. You need to write the full form. Now this arginosuccinate is converted into arginine with the help of an enzyme called as I write the complete name, succinase. Now, here there is a removal of one compound which is known as fumarate. It is very important to remove this one because the carbon number is going to be fixed now. Okay, now the arginine is converted into ornithine, but there is release of urea over here. So arginine is converted into ornithine with the help, by the removal of urea with the help of enzyme called as same arginase. So this is a simple urea cycle. Again, I'm giving a summary. Carbon dioxide and ammonia make a carbonyl phosphate that reacts that combines with ornithine to make arginosuccinate that converts into arginine with the removal of fumarate. Then the urea is removed and arginine is again converted into ornithine. Ornithine again combined with the same carbon dioxide ammonia, and further the reaction goes on. So this is a simple urea cycle. Now, what are the functions of liver? Number one, regulation of blood composition. As we have discussed earlier, that these are responsible for making the red blood cells and the decomposition of red blood cells. Number two, it controls the glucose concentration in our blood. Um, the extra glucose is then converted into glycogen if required. It controls the amino acid level. If there is more amino acid, definitely that is going to convert into urea. It controls the lipid levels. It removes the toxins. It removes the unwanted proteins. For example, the hormones don't need to stay in our body for a longer term. They need to be decomposed, and that is done by the liver. For example, if you have a stress, then you cannot live with that stress for a whole life. That needs to be removed. Okay, then it produces bile from RBC breakdown. There is a synthesis of plasma proteins, then the cholesterol, and then store vitamins. And the last one is the synthesis of the fetal RBCs. It's related to red blood cells only. Then the next one is the carbohydrate control. Now, as we have discussed, that extra glucose is then converted. So first of all, exhaust sugar from digestion converted into glucose, and the regulation of glucose under control of insulin and glucagon. If there is more glucose, then that glucose is converted into simple glycogen that is stored in our body. Then lipid control, only a small amount of glucose can be stored as glycogen. And excess is converted to lipid for storage anywhere. So if you think you are not taking oil and only taking glucose or carbohydrate, or even then that is converted into lipids and stored in your body. So cholesterol is used in many cell membrane. It's a very important part. So every cholesterol is not bad. We have a good cholesterol as well. So excess cholesterol is removed by the liver and excreted by bile. Hormone control, hormone needs to be broken down after they have delivered their message. um, the protein hormones are deaminated and the steroid hormones are excreted in bile. Then the toxin controls, apart from urea, there are other toxins as well. For example, alcohol and certain type of drugs. that also include medicines that need to be detoxified. So liver cell contain enzymes to detoxify these substances. How does the alcohol detoxify? Number one, ethanol is converted into ethanol with the help of dehydrogenase, first of all, and NAD is reduced to NADH. Then ethanol is converted into ethanoic acid with the help of ethanol dehydrogenase. And again, the NAD is converted into NADH or reduced to NADH. This again converted into acetyl coenzyme A. And acetyl coenzyme A is an important part of our Krebs cycle. So it helps in the respiration. So we'll discuss the structure later with the different forms and the other video. Stay tuned. Till then, goodbye.